Biology 1107, Presentation Number 3, Organic Chemistry. Organic chemistry is the chemistry of the compounds of life. We take a look at organisms. They are commonly composed of carbohydrates, compounds called lipids, which would include fats and oils and proteins, and nucleic acids. Those are the four major classes, and we'll be talking about those in a little bit. But those compounds are commonly referred to as biomolecules because they occur in life. These molecules are ultimately composed of covalently bonded carbon atoms. You'll see the molecule illustrated at top right. Now I see 4 carbon atoms with associated hydrogen atoms. That is a classic example of an organic compound. Now, many of the organic compounds that we find in organisms are polymers or long chain molecules made up of individual units called monomers. Not all of them, but many of the carbohydrates and definitely the proteins and definitely nucleic acids qualify as polymers. So please remember, a polymer is a long chain molecule composed of repeating units, and each individual unit is called a monomer. Starch is a great example. You'll see it illustrated at the bottom of the slide. This starch molecule is composed of a series of hexagonal rings, and each of those hexagonal rings is a glucose molecule. So I've got glucose monomers that are linked together to form a long chain molecule. That thing we call starch, that is a classic example of a biomolecule found in plants and found in the plant materials that you and I eat, like rice, and potatoes, and grains, and things like that. Well, if we're going to call these biomolecules and the molecules of life, what do they do for living organisms? What functions do they provide? Well, oftentimes biomolecules contain stored energy, and the stored energy is found in energized electrons found in those covalent bonds. Carbohydrates and fats are good examples of energy-containing compounds. Now, I'll make a comment here, and that is when we think about energy contained in a compound, energy is measured as calories. So any calorie-containing compound is an energy-containing compound. And so I see carbohydrates, which include sugars and fats over there, and obviously if we can deconstruct a sugar molecule or deconstruct a fat molecule, we can extract the energy from those. Now, on the other hand, some biomolecules are structural. They're used to build things. Once again, carbohydrates and fats and proteins fall in those categories, and some are functional. They do things for us. Proteins can function as enzymes, that is, they catalyze reactions, or proteins can function in muscle contraction, or nucleic acids function in storing genetic information. So like I say, we are going to focus on 4 major classes of organic compounds: carbohydrates, lipids, proteins, and nucleic acids. And the fact of the matter is in our own diets, what qualifies as food molecules, which are the molecules that contain stored energy, those are the same thing. You consume food, you're consuming organic compounds. How we use those organic compounds or appropriate those organic compounds largely is determined by our metabolic needs. If we need calories, we use those organic compounds for calories. If I need building blocks for making more proteins, for example, I will use those proteins as a source of amino acids for making more muscle tissue. Or if I need to insulate my nerves for the purposes of transmitting the electrical information that travels through my individual nerve cells, I'm going to use fat to provide me with that insulation around those highly specialized cells and structures. Let's take a look at the anatomy of a typical organic molecule. What's it made of? How's it put together? What it looks like. A lot of what an organic molecule does has everything to do with its structure. So we mentioned that organic compounds are ultimately composed of covalently bonded carbon atoms. Now those carbon atoms constitute what we're going to call the carbon skeleton, which is the framework upon which the rest of the molecule is built. Looking at the illustration of glucose at left and a related sugar, fructose, at right, you will see that the compound is composed of a linear skeleton of carbons that are covalently bonded to one another. In some molecules, that carbon skeleton might be branched. In the case of glucose and fructose, in nature, those sugars oftentimes are formed as ring structures, and sometimes there might be double bonds present in the structure as well. Regardless, any organic compound begins with a carbon skeleton of covalently bonded carbon atoms. Now, what we put on that carbon skeleton are functional groups. A functional group is a small arrangement of atoms that confers properties on the overall molecule. If you take a look at our sugar molecules up there, you'll see little OH groups, and those are hydroxyl groups. Those hydroxyl groups confer properties on the overall molecule. If you take a look at the amino acid, which is illustrated at the bottom left, you will see a blue nitrogen and 2 hydrogens. That's an amino group. On the right-hand side of that molecule, you'll see a carbon double bonded to an oxygen in red and single bonded to a hydroxyl group, which also includes a red oxygen. That is called a carboxyl group or an acid group. So I guess I could assume that I call it an amino acid because it has an amino group and an acid group, and it has a certain behavior because of those functional groups. Functional groups are key to the unique properties of these organic compounds. Now, on our next slide, we're going to summarize what is known about some functional groups. There are, I believe, 6 of them on the next slide, but I only want you to know 5. So I've got a list here. I need you to be able to recognize what is a hydroxyl group, what is a carbonyl group, what is a carboxyl group or acid group, what is an amino group, and what is a phosphate group. And you want to be able to recognize them by looking at a molecule, but also, please, I want you to know some of the added information that we're going to find on the next slide. And we'll run through that very, very quickly. Okay, here's our chart of common functional groups. They're listed down the left. So I do need you to know hydroxyl, I do need you to know carbonyl, carboxyl, amino, and phosphate. The only one I do not want you to know is thiol. Now, my point in covering this at this moment is to make sure there are some things about this chart you need to know. I'm not going to cover the details, but I do need to give you the key to understanding the information contained in this chart. So let's take a look at the column labeled structure. You'll see a letter R. The letter R is essentially code for the rest of the molecule. If I've got, for example, ethanol, which is an organic alcohol that has an OH group on it, I don't worry about the other carbons. I'm worried about the OH group. So the R indicates the rest of the molecule, and the OH indicates the functional group. Same thing with the carbonyl group, same thing with the carboxyl. You will see a column labeled properties. A lot of these properties we already mentioned before, but I just need you to remember polar means charged, nonpolar means not charged. Now, the far majority, as a matter of fact, I see all of these functional groups are actually polar. If you're polar, because you're charged, you're generally attracted and attractive to water, you're hydrophilic. So that's something you need to know for the column on properties. Common functions kind of gives me an idea of what these functional groups do for the compound that has them and where I might find them. So, for example, if I'm looking at an amino group, I understand amino groups are found on amino acids. I know that amino acids are found in proteins. So if I'm looking for a protein, I'm looking for amino acids. And the name amino acid gives me a really good clue as to what this organic compound looks like. It's probably a carbon. It's probably got an amino group on one side, and it's got an acid or carboxyl group on the other side. Now, as far as the amino acids are concerned, what's really helpful is those amino groups and carboxyl groups are critical for linking those amino acids together to making proteins. And we'll see that not too long from now. So make sure you know those 5 functional groups and make sure you're able to recognize them if you are looking at an organic structure. Let's turn our attention to these 4 classes of organic compounds. I will begin by saying that there are plenty more classes of organic compounds out there. But the ones we find specifically found in life are these 4 groups. The other ones account for plastics and other aspects of organic chemistry that don't necessarily involve living organisms. But in living organisms, we find carbohydrates, we find lipids, we find proteins, and we find nucleic acids. And our job is to identify and describe these different classes and do further analysis of what they look like and what sorts of compounds we find in each group and what they do for us. So let's take a look at carbohydrates first. Carbohydrates are compounds that we commonly refer to as sugars or starch or fiber. And all carbohydrates have the same basic chemical formula. You will see CHO equals 1 to 2 to 1, meaning that any molecule of carbohydrate contains carbon atoms, hydrogen atoms, and oxygen atoms, always in a ratio of 1 to 2 to 1. Now, many of the carbohydrates that we find in biological situations are polymers or long-chain molecules. But those long-chain molecules are composed of individual units called monomers, and monomers in the carbohydrate group are what we're going to call monosaccharides. Now, mono means one, and saccharide means sugar. So these are single unit sugars, and sometimes we call them simple sugars as well. You take a look at the right-hand side, you will see an arrow pointing to a structure that looks like a hexagon. That is glucose, and you'll see another molecule that looks like a pentagon, and that is fructose. Incidentally, and we'll see this on another slide, that glucose and fructose, even though they look differently, have the exact same chemical formula. Glucose and fructose are both C6H12O6. And once again, that fits the bill. C6, 6 carbons, H12, 12 hydrogens, O6, 6 oxygens. It's a ratio of 1 to 2 to 1. Now, if we take 2 monosaccharides and link them together, we get a disaccharide. And the most common disaccharide, at least the disaccharide you're most familiar with, is something called sucrose. That's common table sugar. So I've got a glucose linked to a fructose, and that's a 2-unit sugar. That's a disaccharide. That's what you find on your sugar frosted flakes. That's what you put in your tea or in your coffee. Now, the linkage between those 2 monosaccharides is called a glycosidic bond. I will need you to know a glycosidic bond is a link found between 2 monosaccharides in the construction of a disaccharide, or these are also links found in the construction of a polysaccharide like starch below. So what's a polysaccharide? Well, a polysaccharide would be many monosaccharides linked together into a true polymer. Multiple unit sugars. You'll see starch down there. We saw a picture of starch on a previous slide. It just looked a little bit different. But I see individual glucose molecules in a series, and I see this funky little bond with the oxygen in the middle of it, a glycosidic bond linking each of those sugars into that long polymer chain. No monosaccharides, no disaccharides, no polysaccharides. So what else do you want to know about carbohydrates? Well, most carbohydrates are water soluble. Carbohydrates are oftentimes composed of polar covalently bonded carbon atoms. Now, you might remember a polar covalent bond is one where the electrons are shared unequally, and this means there might be little positive and little negative charges all over that sugar molecule. As a consequence, that sugar molecule tends to dissolve in water. Carbohydrates, as a rule, are polar compounds for that reason. Carbohydrates include fiber, which are compounds common to plants, and those are insoluble carbohydrates. They will attract water, but generally they don't dissolve. Monosaccharides are classified into types based on the number of carbons they have, so we could talk about hexose sugars or pentose sugars or triose sugars. As a matter of fact, DNA consists of pentose sugars. The ribose or the deoxyribose found in a DNA molecule is a pentose or 5-carbon sugar. On the other hand, glucose and fructose, the constituent molecules found in sucrose, are hexose sugars. Now, I mentioned glycosidic bonds. Glycosidic bonds are linkages between monosaccharides to form a disaccharide or between monosaccharides forming polysaccharides. I'm going to spend a bit of time here talking about how glycosidic bonds are formed. And the thing is, it's important from the standpoint that not only are these reactions that form glycosidic bonds characteristic of carbohydrates, but they will also be characteristic of linkages between amino acids in the formation of proteins as well. So let's take a look at the illustration below, and we'll see the words condensation reaction and hydrolysis reaction. Now, the name condensation implies the fact that we are going to be producing water in the process of the reaction. Water is going to appear. As a matter of fact, you will see the illustration at top there, and it's relatively small, but you'll see two molecules that are about to be linked together. And those molecules are going to be linked together through the formation of a glycosidic bond, but you will see in the yields arrow that connects the reactants on the left to the product on right, you will see the formation of a molecule of water. Furthermore, you will notice that an OH group and a hydrogen are highlighted on the left. And so what we're going to be doing is we're going to be removing the OH and the hydrogen, forming water in the process. And when that OH and that hydrogen are removed, that opens up bonding sites to create the linkage between the two. That is called a condensation reaction. Please make sure that you note that condensation reactions make linkages. A condensation reaction makes a linkage through the production of water. Condensation reactions are sometimes called dehydration synthesis reactions because what you're doing is you're actually removing water from the two molecules as you create the linkage. Condensation reaction, dehydration synthesis reaction, those are two synonyms. So what about breaking these glycosidic bonds? Well, if you break a glycosidic bond, that is called a hydrolysis reaction. And once again, a hydrolysis reaction is not limited to carbohydrates. Anytime you break a bond, it's conceivably called a hydrolysis reaction. Now take a look at the name. Hydrolysis, hydro, meaning water, lysis means to break. So water is our agent in breaking these bonds. Take a look at the illustration at the bottom. You will see our glycosidically linked disaccharide molecule. You will see the addition of water at the yields arrow, and you will see our two separate individual monosaccharides at right, and I have replaced that glycosidic bond with the OH on one monosaccharide and hydrogen on the other. So we think about condensation and dehydration synthesis reactions of flip sides of the same coin. I can make linkages, I can break linkages. Linkages are made through condensation reactions, sometimes called dehydration reactions, and linkages are broken through hydrolysis reactions. And those terms, dehydration, condensation, hydrolysis can be used to identify the making and breaking of bonds in any organic class of compounds. Carbohydrates, as far as humans go, are our body's first priority as a food molecule. You might want to underline food molecule and remind yourself that a food molecule contains stored energy, and that energy can be quantified or measured as calories. Now, I'm not going to get into the actual quantity of what a calorie is, but make sure that you equate that word calorie with energy stored in a food molecule. If we are presented with an opportunity to extract energy from a carbohydrate, from a fat, from a protein, we will extract that energy from the carbohydrate first. Now, when you break down a sugar molecule, and it occurs in our individual cells through a process known as aerobic cellular respiration, what we do is we get carbon dioxide, which represents the carbon atoms that constructed that food molecule in the first place. We get electrons, because remember, those are energized electrons contained in the molecule, but we're going to extract the electrons, and we're going to use those electrons to construct a molecule of water. But we're also going to get some energy. And it's the energy on those electrons are going to be used to make a form of cellular fuel that you and I know as ATP. So we're not going into detail about cellular respiration right now, but in anticipation of a future presentation, I do want you to make sure that you memorize this reaction found at the bottom of the slide. A molecule of glucose broken down in the presence of oxygen yields 6 molecules of carbon dioxide, yields 6 molecules of water, and yields about 38 molecules of ATP, which is a form of cellular fuel. Information that we're going to cover not too long from now. Carbohydrates, as far as humans go, are our body's first priority as a food molecule. You might want to underline food molecule and remind yourself that a food molecule contains stored energy, and that energy can be quantified or measured as calories. Now, I'm not going to get into the actual quantity of what a calorie is, but make sure that you equate that word calorie with energy stored in a food molecule. If we are presented with an opportunity to extract energy from a carbohydrate, from a fat, from a protein, we will extract that energy from the carbohydrate first. Now, when you break down a sugar molecule, and it occurs in our individual cells through a process known as aerobic cellular respiration, what we do is we get carbon dioxide, which represents the carbon atoms that constructed that food molecule in the first place. We get electrons, because remember, those are energized electrons contained in the molecule, but we're going to extract the electrons, and we're going to use those electrons to construct a molecule of water. But we're also going to get some energy. And it's the energy on those electrons are going to be used to make a form of cellular fuel that you and I know as ATP. So we're not going into detail about cellular respiration right now, but in anticipation of a future presentation, I do want you to make sure that you memorize this reaction found at the bottom of the slide. A molecule of glucose broken down in the presence of oxygen yields 6 molecules of carbon dioxide, yields 6 molecules of water, and yields about 38 molecules of ATP, which is a form of cellular fuels. Information that we're going to cover not too long from now. If you take a close look at this particular slide, it documents some content that we've already mentioned. Probably the most important bit here is something that I did not fully explain. I did mention that glucose and fructose have a chemical composition that is similar to one another. Matter of fact, identical to each other. The most commonly found sugar in all of life is glucose. Fructose is common to plants. It's commonly referred to as fruit sugar, but when we put those two compounds together, we get a molecule of sucrose, or common table sugar. Fructose and glucose are isomers. They have the same chemical formula, C6H12O6. In other words, they're both hexose sugars, but they look different. Now, just because they look different doesn't mean they have different chemical formula. So if you take a look at the hexagonal structure of glucose, or you take a look at the pentagonal structure of fructose, and spend some time counting the number of carbons, you will note that there are 6 carbons in the glucose and 6 carbons in the fructose. They're just arranged differently. So sucrose is the result, a disaccharide composed of glucose and fructose, ultimately created by a glycosidic linkage between the two. And if you take a look at the illustration, you'll see that water is being removed in combination from the glucose and the fructose. So that reaction qualifies as a dehydration synthesis or a condensation reaction, creating that glycosidic linkage between glucose and fructose. And I know it's like flogging a dead horse because we've covered that information before, but one more time won't hurt anybody. So what we'll be doing on the next 3 slides is documenting the 3 major polysaccharides that are actually critical to life. And those include starch, cellulose, and glycogen. Our first slide documents starch. Starch is how plants store their food energy. We will be talking more detail about photosynthesis later in the semester, but we already know that photosynthesis is a process how plants make their own food molecules. Specifically, it's how a plant takes carbon dioxide, water, and the sun's energy to make a molecule of glucose. Now, a plant could conceivably use that glucose to maintain life, to fuel growth, or to fuel reproduction. But in many cases, the plant might also want to hold on to that glucose and store it. Because even though a plant makes its own food molecules at nighttime, it does not have that capability. And most of us realize that during the winter, and some plants don't have leaves, and some plants aren't capable of doing photosynthesis, where are they going to get their food molecules? Well, plants store those glucose molecules in the form of starch. Starch is our way of storing food energy if you're a plant. Now, starch is actually a mixture of 2 different compounds. The starch that you find in cereal, the starch that you find in rice, the starch that you find in potato is a composition of 2 different chemical compounds. Each of these compounds is a polymer of glucose units. One is called amylose. Amylose is an unbroken chain of glucose units. And you'll see on the illustration here, it is a linear chain. In nature, what will happen is the string of glucose molecules will actually coil into a helix. Now, there's a second compound. It's called amylopectin, in which case the glucose polymers are highly branched. And those 2 molecules together constitute what we call starch. The same starch you find in bread, the same starch you find in all of these foods that you consume that contain a significant amount of stored energy. Our next polymer, or polysaccharide of glucose, is cellulose. And is the most common form of fiber that we find in our diets. And that's because cellulose is the primary component of the cell walls of plant cells. You consume green beans or peas or lettuce on a salad or cabbage. All of those materials are composed of plant cells, and those plant cells are reinforced with external plant cell walls, and those cell walls are made of this stuff called cellulose. Cellulose consists of long, unbranched chains of glucose molecules, but you'll notice in the illustration that those glucose molecules are cross-linked. And this constitutes what amounts to being a compound that, while at one level seems to be energy rich, we as humans do not have the enzymes that are capable of digesting cellulose. Now, there are enzymes out there in the world of biology that can. For example, if you take a look at termite in its gut has little teeny tiny little single-celled protists that are actually living in a termite's gut, and those organisms and many fungi and many bacteria do make an enzyme that could be conceivably called cellulase that breaks down cellulose into individual glucose molecules, which are a significant source of energy for those organisms. So I'm taking a look at a plant. So I'm going to realize that cellulose, even though it's the most common form of fiber found in plants, it's not the only game in town. Other fiber compounds found in plants are lignin, which is a woody material, and hemicellulose and pectin, which are substances that kind of help glue plant cells together. But the bottom line is, even though these compounds appear to be energy rich because they're loaded with glucose, because of their structure, they are essentially non-digestible. So as far as humans are concerned, fiber is pretty much a functional thing, not a digestible thing. Meaning, as functional, it will aid in the movement of fecal material through the intestines. It will provide food materials for the bacteria living in our intestines, but it is not a significant source of calories. It's not a source of calories at all for us. So we have been covering what I would call the biological big three polymers. Those would be starch, cellulose, and now let's take a look at glycogen. If you take a look at the illustration on the left-hand side of the slide, you will see that glycogen looks very, very similar to amylopectin that we became introduced to in talking about starch. Glycogen is a branched polymer of glucose. Glycogen is how animals store food energy. In other words, while plants store starch, animals store glycogen. And in humans and in most animals, when you have excess blood sugar that generally arises after eating, and you're not using that blood sugar for your immediate needs, you store it. And in humans, you store it in one of two different places. You store it in your muscle cells, or you store it in your liver. In any event, it provides you some ready fuel that is available should you need it. One of the reasons why we store glycogen in our muscle cells is for that exact reason. If an opportunity arises for you to need to contract your muscle tissue, that takes energy. What better way to provide that energy by having an already in place energy containing molecule in your muscle tissue? Now, on the other hand, in our liver, we store this stuff called glycogen, and that glycogen is ultimately made from excess blood sugar. So when you eat, your blood sugar levels go up. You are going to release an hormone called insulin, which is going to give a message to your liver, among other places, to basically make glycogen. So the liver is going to undergo a process called glycogenesis. Now, the word glycogenesis means creating glycogen. So what we're going to do is we're going to take the blood glucose levels, and you're going to bring them down. And as we bring those blood glucose levels down, the glucose we are removing by the liver will be linked together to form glycogen. That glycogen will be stored in the liver for events and times when you might not have enough blood sugar. So we're talking about our schedule on a daily basis. You eat breakfast, your blood sugar levels go up. Those blood sugar levels go back down because your liver will be storing excess glucose as glycogen. You eat lunch, blood sugar levels go up, and they go back down, in part because your liver is removing that glucose and storing it as glycogen. You eat dinner, same thing. Blood glucose levels come down because the liver is constructing glycogen. That's called glycogenesis. Now, in between meals are those times when you need that blood sugar, because your cells need a supply of blood sugar regardless of whether you're eating or not. So you've eaten breakfast, your blood sugar levels go up, they come back down. Maybe it's 10 o'clock, maybe it's 11 o'clock. It's almost time for lunch, but the fact of the matter is your blood sugar levels are really low. Your cells need blood sugar. They don't care whether you've not eaten or not. You still need that blood sugar. Those cells are clamoring for blood sugar. So what happens is in the liver, that glycogen can be dismantled to release glucose as we need it. And that process is called glycogenolysis. Glycogenolysis. Olysis means to break, to split. So you'll notice in my little boxed item down here, I've documented what I've just been talking about. You eat, your blood sugar levels go up. After the meal, glycogenesis takes place, your blood sugar levels go down. But you're storing glycogen in your liver. So if there's no eating going on, your blood sugar levels go down. And in between meals, it might get to that point where you need to release some glucose. And that process is called glycogenolysis. And your liver dismantles that glycogen, releases that glucose, and your blood sugar levels go back up, which means your cells can be supplied by that glucose as they need it. So let's turn our attention to a fairly broad class of organic compounds called lipids. As a rule, these are compounds that are nonpolar. So as molecules, they are constructed of nonpolar covalent bonds. They are hydrophobic. They do not mix with, nor do they dissolve in water. These compounds are not polymers. It's one of the few groups of organic compounds that don't exist as polymers. Many carbohydrates do, proteins do, nucleic acids do, but lipids do not. I said it was a fairly broad class, so we are going to examine compounds called triglycerides, which include what you and I would call fats and oils, phospholipids, sterols, and waxes. Let's take a look at our first category of lipids, triglycerides, compounds that we commonly call fats and oils. Now, I do not want you to be overwhelmed with the chemical structures here. We're not going to memorize any chemical structures, but what we are going to do is focus on some aspects of chemical structures that will explain to us why these molecules are identified as what they are and how they work. Let's first take a look at the basic structure. Take a look at the name triglyceride. It's three of something. We're going to see it's three fatty acids that are bonded to a molecule of glycerol. Take a look at the structure at right. The glycerol molecule is a 3-carbon alcohol, and it's highlighted by the red box. Each of those carbons is going to be a place where we will bond a fatty acid. The fatty acids are illustrated to the right of the glycerol. They're highlighted in the orange boxes. And fatty acids, as a rule, are long hydrocarbon chains. Now, the word hydrocarbon chain implies lots of carbons and lots of hydrogens. If you take a look immediately below our generalized view of the triglyceride at top, you will see that these fatty acids come in two different flavors. There are two different types. There are saturated fatty acids and there are unsaturated fatty acids. This is what I need you to know. The first thing is a saturated fatty acid is a fatty acid that is characterized by carbons that are all single bonded. Take a look at that structure below that's illustrated as a saturated fatty acid, and you'll see those carbons are all single bonded. There is a lot of them. And every single one of those carbons in that chain has two hydrogens also bonded to it. So I could say that that particular fatty acid has the maximum number of hydrogens surrounding that linear chain of carbons. That's why we say it's saturated. Maximum number of hydrogens means that is a saturated fatty acid. In contrast, look at the unsaturated fatty acid below, and you see there is a double bond smack dab in the middle. That double bond does two things for the fatty acid. First off, what it does is it reduces the number of hydrogens found in a chain. So if this chain has less than the maximum number of hydrogens, we can say it's unsaturated. The other thing it does is the double bond creates a bend or a kink in the actual chain. So saturated fatty acids, all single bonded, linear chains. Unsaturated fatty acids possess a double bond, and the chain is kinked. Now, it's the fatty acids found in the triglyceride is what really determines the molecule's properties. Every single triglyceride consists of one glycerol molecule and 3 fatty acids, but it's the nature of those fatty acids that really determine that molecule's characteristics and its function. So let's take a real practical look at what that might mean for the triglyceride. We're going to compare what you and I commonly call fats and oils. Now, fats are what we would call triglycerides that are saturated triglycerides because they're composed of saturated fatty acids. I'm understanding I can use that word saturated to describe the fatty acids, but I also can use it to describe the overall triglyceride molecule. At right, you will see a ball model of a saturated triglyceride. And I see 3 red balls representing the 3 oxygens on the 3 carbons of the glycerol molecule, but trailing to the right of those, I see 1, 2, 3 very linear fatty acids. And I see a close-up view of the fact that those fatty acids are, in fact, single bonded. Now, what I will remember is that a single bonded fatty acid is a linear fatty acid. Now, these saturated triglycerides tend to be solid at room temperature. That's why we call them fats. The word fat is associated with some compound that tends to be solid at room temperature, and the only way the darn thing becomes liquefied is if you add heat, at which point it melts. Now, one of the reasons why this compound tends to be a solid at room temperature is because the molecules, because they're linear, pack very, very densely. Think about a box of toothpicks. And the toothpicks are all really, really straight. You can cram 100 toothpicks into a fairly small box, and they pack very, very densely. Hold that thought because we're going to compare that to the unsaturated triglycerides next. So to summarize, fats are solid at room temperature. They are saturated triglycerides. They are composed of saturated fatty acids. They are, as a rule, solid at room temperature because of the dense packing of those linear molecules. And the last bullet point is this. As a rule, fats in general come from animal sources. There are some exceptions, but essentially, for the most part, saturated triglycerides come from animal sources. Now, in contrast, let's take a look at oils. Oils are triglyceride compounds that are liquid at room temperature. The reason why they're liquid at room temperature is they are ultimately composed of unsaturated fatty acids. So therefore, an oil is an unsaturated triglyceride. I'm noting that I can use the word unsaturated to identify the triglyceride, but I can also use it to identify the characteristics of the fatty acids. Take a look at the illustration at far right. I see the same basic structures before. I see the glycerol molecule. I see 3 fatty acids. But the big difference is in this illustration, the fatty acids are kinked or bent, and the inset tells me that the kinks are places where double bonds exist. As a consequence of being kinked, the fatty acids make it impossible for the triglyceride molecules to be able to pack very densely. As a consequence, instead of being a solid at room temperature, these unsaturated triglycerides are liquid at room temperature. And as a rule, these oils, or these unsaturated triglycerides, come from plant sources. There are, of course, exceptions to that, most notably fish oil. If you take fish oil capsules as a dietary supplement, that is a triglyceride that comes from an animal source but is liquid at room temperature. Make sure you know the chemical difference between what are fats and oils, and make sure you know the reasons why they are either solid at room temperature or liquid at room temperature. Triglycerides are an important part of our diet, but what I want to do right now is I want to focus on one aspect of that importance, and that is the fatty acids. When you and I consume fats or oils, we digest those fats or oils through a process called lipolysis. Lipolysis is going to involve certain enzymes that actually break the bonds between the glycerol and the fatty acids. Those fatty acids can be a source of energy. We've talked about cellular respiration, and we understand that cellular respiration can process glucose, carbohydrates, but we can also process fatty acids as a source of energy as well. But more importantly, fatty acids can be building blocks for making other compounds, including triglycerides or even phospholipids that are going to be important in constructing our own cell membranes. But there's a bunch of other things they can do as well. Let's take a look at two basic categories of fatty acids. There are non-essential fatty acids and essential fatty acids. The basic difference between the two is that non-essential fatty acids can be constructed in our own bodies from other molecular components. In other words, non-essential fatty acids do not have to be present in our diets in order for us to benefit from them. If we are lacking a non-essential fatty acid, we can just make it ourselves from some other molecular component. On the other hand, there are essential fatty acids, and those fatty acids absolutely must be present in our diet because we do not have the capability of making them on our own. Now, the essential fatty acids are therefore dietarily very, very important, and they come in two general types. And you've probably heard of them before. They're called omega-3 fatty acids and omega-6 fatty acids. The names have something to do with the presence of a double bond in the fatty acid and how far away from the end of the chain it happens to be. It's a kind of a detail you don't have to worry about. However, there is a difference in terms of what these fatty acids do. Omega-3 fatty acids, which you generally find in fish oils, have an anti-inflammatory characteristic. They prevent inflammation and they lower LDL levels in the blood. Now, LDL stands for low density lipoprotein, but most people simply refer to LDL as bad cholesterol. It's the cholesterol in your body that basically triggers things like atherosclerosis and heart disease and things like that. So, if you want to prevent those LDL consequences, take an omega-3 supplement because it has those anti-inflammatory properties, but more importantly, lowers those LDL levels. Of course, the alternative is just don't eat so much fat. However, let's take a look at omega-6 fatty acids. In contrast to omega-3 fatty acids, omega-6 fatty acids, which primarily come from plant seed oils, so you may have heard of flaxseed oil as an example, these are compounds that actually do have some inflammatory properties. That doesn't mean they're bad. It just means that they have some negative aspects that you might want to be aware of. I mean, I, for one, while I would take an omega-3 fatty acid supplement, I would stay away from omega-6 fatty acid supplements, although there are some fatty acid supplements that will be represented by a mixture of the two. But the thing is the omega-6, yes, while they are inflammatory, they are building blocks for critical molecules in our bodies that are called eicosanoids. Don't have to worry about that word, but these are substances that are important in our perception of pain. They function an awful lot like hormones, and they are of critical importance. So bottom line, what do I need you to know? I need you to know that lipolysis is the term for the chemical breakdown of triglycerides, and they result in liberating these fatty acids. You also need to know that fatty acids come in two different types: the non-essential fatty acids versus the essential fatty acids. And then finally, make sure you know the specific difference between what are omega-3 fatty acids and omega-6 fatty acids, two classes of the essential fatty acids in our diet. While triglycerides are extremely important and dominate our understanding of lipids, there are a few other less well-known categories of lipids, and one of those is the phospholipids. Now, while the phospholipids are less well-known, they are by no means any less important. Let's talk about these unique lipids called phospholipids. The first thing I want to observe here, and it's not specifically mentioned in the bullet points, but you can see it from the illustration, and that is this. A phospholipid is very similar in construction to our triglycerides. So what I would like you to do is, next to the slide, you write down somewhere that a phospholipid consists of three different components. Number one, you want to write down a phospholipid has a glycerol molecule, which is what we saw in the triglycerides. Number two, a phospholipid has two fatty acids. Not three, two. And then number three, a phospholipid has a phosphate group. So let's take a look at the ball model that I have to the right-hand side of this slide. I mean, that ball model is actually accompanied by a standard chemical formula structure, but I want to look at the ball model. You see the ball model has two regions of the molecule highlighted. The region at the top with the blue shaded circle around it is the head portion of the molecule, and below that is the tail portion of the molecule, which is shaded in orange. At the intersection of the two, just below the blue circle, you will see a series of red oxygen atoms. That's our glycerol molecule. So our glycerol molecule is smack dab between the head portion and the tail portion of the molecule. Now, looking at the tail portion of the molecule, you will see, and they are easily seen, two fatty acid chains. Above the glycerol molecule is where we find our phosphate group. That's why this thing is called a phospholipid. Now, the phospholipid has a charge. It's a polar molecule, at least in part. If you take a look at the phosphate group, that phosphate group up there at the top of the molecule is going to have a negative charge. Because of that, the phosphate group will be hydrophilic. Because of that, the phosphate group portion of the molecule, the head portion of the molecule, can be described as being polar. So I see the word hydrophilic up there. You might want to scribble next to that the word polar, just to make sure that you know what you're talking about. Beneath that, you will see the two fatty acids, which are essentially every bit as nonpolar as the fatty acids were in triglycerides. Hence, I've got the word hydrophobic. You could go ahead and write the word nonpolar next to that. So what I got here is a molecule with two parts. I've got a molecule that technically has a split personality. The head portion of the molecule behaves completely differently from the tail portion. In our bullet points, you will see the word amphipathic. That is an adjective. It is a word that describes a molecule that has two distinct chemical characters. Put another way, amphipathic is a word that describes a molecule that has a split personality, like our phospholipid here. One part of the molecule is polar, one part is nonpolar. And your job is to remember that the head portion is polar and the tail portion is nonpolar. So now this particular molecule has a very unique behavior when it occurs in aqueous solution. Aqueous solution means water. If you put phospholipids in water, they will always form a bi-layered arrangement. If you take a look at the illustration immediately below our diagram of the phospholipid, you will see what looks like a bunch of red balls with tails. And there's 2 rows of them. Those are phospholipid molecules, and they arrange themselves in this manner. And this is what happens. What happens is the ball portions of those molecules illustrated there are the heads. And you'll remember the heads are hydrophilic. They're water-loving. So what you could do is you could draw itsy-weetsy, teeny tiny little water molecules on the extracellular side of that bilayer and teeny tiny little water molecules on the intracellular side of that bilayer. That's where the water molecules are going to be located, and this is one of the reasons why the heads are located on the outside of the bilayer, because they are much more chemically comfortable, let's put it, being close to water molecules. On the other hand, the tail portion of the molecule can't stand water. So therefore, one of the reasonings behind forming this bilayer is that all the tail portions of your molecule are located at the center of the bilayer, and all water is excluded. So while you've got water molecules above the bilayer in this picture and water molecules below, there are absolutely none where those tails are. As a consequence, the molecule arranges itself so it is much more, let's put it, chemically comfortable with the water in which it's existing. Now, when you create this bilayer, that creates a physical structure that cells have appropriated. It is the primary structure of biological membranes. It becomes an interface, and it's why you see the word intracellular on one side and extracellular on the other side, because a biological membrane is like your cell membrane. It's what separates the cell from the environment around it. So phospholipids are similar to triglycerides in their structure, but the big difference is one part of the molecule, the head, is polar, hydrophilic. The other part of the molecule, the tail portion, is hydrophobic. It's nonpolar. As a consequence, this is a molecule with a split personality. It is amphipathic, and it has a very specific behavior in water, in aqueous solution, that lends itself very well to be the primary structure of biological membranes. Cell membranes, nuclear membranes, mitochondrial membranes, chloroplast membranes, every single type of membrane you find in any cell on the face of the planet, and there's lots of them, phospholipids are the number one structural component. Our third category of lipids are sterols. Sterols, or commonly referred to as steroids, bear no chemical resemblance to either the phospholipids or the triglycerides. Sterols have a characteristic structure, and you'll see it repeatedly on this particular slide. It's a 4-fused ring structure. Now, the way to look at these models of chemical structure is every time you see an intersection between 2 lines at an angle, that's where a carbon exists. So you notice we don't have the individual carbons illustrated, but they do exist where every single line meets another in an angle. And so I've got a 4-fused ring structure that constitutes the basic structure of these things called sterols. Now, all sterols in animals are ultimately derived from a molecule called cholesterol. And so you'll see cholesterol illustrated at the top of the slide. That is the source molecule for all of the sterol molecules that you and I have that do an awful lot to control our sexual development, control the response to stress, and basically contribute to our ability to metabolize calcium. Let's take a look at what we've got here. You will see the word estradiol, and you will see the word testosterone. Testosterone is a male sex hormone. Estradiol is also known as estrogen. That is a female sex hormone. You will notice that those two hormone molecules are almost identical in terms of their structure. They both have the characteristic for fused rings that we see in cholesterol. Cortisol, you'll see below that, is the stress hormone, and then you'll see vitamin D. Now, vitamin D is something that actually helps us to absorb and metabolize calcium. So one of the reasons why they package milk with vitamin D and call it vitamin D milk is because vitamin D is known to actually improve the absorption of that calcium. So make sure that you know the basic structure of sterols. These are still lipids, they're still hydrophobic, but their structure does not resemble the two classes we have just previously looked at. Know the representatives, and we just got one more group to look at, and let's see what that is. Our final category of lipids are waxes. Doesn't seem to be a lot of content on this slide, but nonetheless, waxes are important compounds, if only from the standpoint that we think about them as being extremely hydrophobic or water repellent compounds. These are very, very long chain molecules. As a matter of fact, so the illustrations here do the exact same convention that we saw earlier. I'm neglecting to include the carbons. Makes life a lot easier. Every single angle between the straight lines is a carbon atom. Large number of carbons, all single bonded, extremely hydrophobic. Now, most waxes are of plant origin. As a matter of fact, should mention this. All plants have a non-cellular layer of wax that coats their leaf surfaces. And one of the reasons this exists is because the plant is living in a desiccating environment and obviously does not want to lose water through the process of evaporation from the leaf surfaces. So the leaf surfaces are sealed against that evaporation by this layer of wax on the plant's leaf surface. That structure, that layer of wax, is called a cuticle, spelled Cuticle. And I know it's not on this slide, but you need to know it. And the name of that wax is called cutin, Cutin. Plants have a layer of wax on their leaf surfaces that prevent those leaves from drying out in a desiccating environment. The wax is called cutin. The layer of wax is called a cuticle. And most waxes are of plant origins. As a matter of fact, if you took a look at a plant, you might notice, for example, a magnolia, the leaves are very, very shiny green. Same thing with hot holly tree. Same thing with most leaves in the plant kingdom. They have some degree of shine, sometimes more, sometimes less. But the shine is an indication of the presence of this cuticle. The only exception I can think of as far as waxes are concerned is beeswax, as a wax that is produced by an animal. And obviously bees generate that wax from the compounds they obtain from the plants in the first place. So at this point, we're finished with lipids. I need you just make sure you know, as a group of organic compounds, it's fairly diverse. It consists of molecules that are extremely hydrophobic, with the exception of the phospholipids. They're kind of got a split personality, and part of that molecule is hydrophilic. But they all have that one thing in common, and none of them is a polymer. Now we're going to turn our attention to our third class of organic compounds in a moment, and that will be proteins. And proteins definitely are polymers. They are arguably the most important class of organic compounds, as we'll see in a minute. This brings us to our portion of the presentation that deals with proteins. Extremely large molecules of incredible diversity. Proteins are molecules that have a specific shape. You'll see an illustration of hemoglobin at right, which is the protein that's found in our red blood cells responsible for binding oxygen. Is critical to our ability to transport oxygen in our bloodstream, and it has a very specific shape, and its function is linked to that shape. And in spite of the fact that they're so big, and in spite of the fact that they're so complicated, every single protein is nothing more than a polymer of amino acids. Now, I'm going to introduce a term here. It's going to be found on a number of the slides, but I'm going to go ahead and introduce it now, and you can write it down, and that is the word polypeptide. Polypeptide. A polypeptide is a strand of amino acids. A polypeptide is a polymer of amino acids. And in general, when you look at a protein, like that hemoglobin molecule, it is a construction of multiple strands of amino acids. That hemoglobin molecule is actually a composite of 4 different polypeptides. So it's an important word because you're constructing a protein out of amino acids, but basically you're going to take those amino acids and you link them into a polypeptide, and that, with some other polypeptides, ultimately will give you the structure of your protein. So let's take a look at amino acids. On our following slide, we'll actually have an illustration that documents the structure, but what we want to know right now is that amino acids are unique nitrogen-containing molecules, and to the right of this information on the slide, you'll see a composite illustration including a bunch of different chemical structures. Now, all the chemical structures appear roughly the same. The only thing that's different is the shaded area, and the shaded area on each one of those amino acids constitutes something that we're going to call the side chain. And we'll see on our next slide that it is the side chain that makes each amino acid unique. Well, there are 20 different amino acids that are found in nature. Now, human beings, we can make 11 of those, and the ones we can make are called non-essential amino acids. We use the same basic definition that we used when we were talking about non-essential fatty acids. Non-essential fatty acids, remember, we could make them on our own. Well, the same thing is true with non-essential amino acids. We can construct these on our own from readily available molecules. If we're low on an amino acid like arginine, all we have to do is find ourselves a suitable organic molecule and use that as our source molecule for building a new arginine. On the other hand, 9 of those 20 amino acids, as far as humans are concerned, are essential amino acids, meaning we do not have the ability to construct them on our own, meaning they have to be found in our diet. And if we don't have the essential amino acid present in our diet, and if that amino acid is necessary for the construction of a protein, you can't make the protein unless the amino acid is present. It's called the all or none principle. And the fact of the matter is, if that protein is necessary to sustain life, then you are going to have an impact on your quality of life. You might get very sick. You might even die if you don't have that protein available. So amino acids are the building blocks for making proteins. Now, in a minute, we'll take a look at the specific structure of an amino acid, and then we'll understand how those amino acids become linked together to form polypeptides, and then how those polypeptides become organized into these large, critically important molecules called proteins. All right, here's our basic amino acid structure, and we've mentioned this not too long ago when we were introducing the notion of functional groups. So here's our amino acid, and it consists of a central carbon, and to the right of that central carbon, I see a carboxyl group, also called an acid group, and to the left, I see in green an amino group. And so that's what gets our amino acid its name, has an amino group and an acid group. And then there's this thing called the side chain, and the side chain is what makes this particular amino acid unique. The side chain can be anywhere from a simple hydrogen to a methyl group, which is CH3, or it could be something really, really long and extravagant. You saw those structures illustrated on a previous slide. If I take a look at the actual ball and stick model, you'll see it to the right, same constituents seen in that model as well. Now, we're going to see in a minute that while the side chain is what confers the specific identity on the amino acid, its function in connecting to and binding to other amino acids is going to have everything to do with that amino group on the left and that carboxyl group on the right. And we'll see that in a moment. All right, so when we link amino acids together, we're actually building a polymer. Now, you might remember the words dehydration synthesis or condensation reaction as terms we used for the formation of glycosidic bonds when we were creating disaccharides or polysaccharides way back when we were talking about carbohydrates. Well, the same terms have application here. I've got an illustration on our slide that shows me 2 amino acids at the top. You will see that the hydroxyl group of one carboxyl group on the left is highlighted, and see a hydrogen of the amino group on the right is highlighted. So what we're going to do is we're going to take those hydroxyl and hydrogen atoms away, and we're going to create something called a peptide bond. So remember, in a condensation reaction, we're generating water, or we can think of it as a dehydration synthesis because we're actually removing water in the formation of this bond. In any event, the bond that is formed has a characteristic appearance. It's called a peptide bond, and you'll want to know that term every bit as important as glycosidic bond. A peptide bond is a linkage between 2 amino acids, and it is generated through a condensation reaction, also known as a dehydration synthesis reaction. Now, I should mention that on the flip side of that, you could conceivably break a peptide bond through a hydrolysis reaction. But at this moment, we're going to make sure that we know what a peptide bond is and how it is formed. This slide is going to give us an idea how we start with something as simple as a handful of amino acids and how we get to something as big and complex as a functional protein. This particular slide documents something that's documented on a whole lot of different websites in a lot of different illustrations. If you don't like this illustration, Google protein structure and you'll come up with 1,001 versions of this same illustration. First off, we are going to begin with acknowledging the fact that proteins have multiple levels of structure. They have what's called primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure is the identity and the sequence of the amino acids linked together by peptide bonds to form a polypeptide. So the illustration at right shows you what looks to be a strand of beads. And if this was a color-coded illustration, that could be a multicolored strand of beads, each color corresponding to one of those 20 different amino acids found in nature. Now, it's important to recognize that those individual amino acids have these side chains, and the side chains vary in terms of how big they are and their chemical characteristics, which means as you run along the length of primary structure, there is going to be some degree of interaction between those amino acids in that polypeptide on account of their side chains. That brings us to secondary structure, because you take that primary structure, that polypeptide can either coil, as I see on the right, or it can fold, as I see on the left. The coiling on the right is commonly referred to as the alpha helix. That's the Greek letter alpha with the word helix, Helix, to imply this coiling. On the left, that's commonly referred to as the beta pleated sheet arrangement. Beta, spelled B-E-T-A, Greek letter for B, pleated sheet arrangement. In any event, it is the individual amino acids along the length of that polypeptide will result either in that folded appearance or that spiral appearance, and that folding or that coiling is referred to as the secondary structure of the polypeptide. There's one more level of structure corresponding to the individual polypeptide, and that is called tertiary structure. And this is a complex three-dimensional folding of the polypeptide. It's as if that strand of amino acids was almost like a piece of wire, and you could fold that wire up into this three-dimensional shape. So you'll see on our illustration at right, in fact, there are places where sometimes we have the helix, sometimes we have the pleated sheet, secondary structure, but the whole shooting match is wadded up on itself in a three-dimensional structure. I'm just going to mention at this point there are certain interactions between amino acids in that polypeptide that are going to literally hold that three-dimensional structure in place. I like to think of them as like pinning points. There's little places where one amino acid will be attracted or connected to another amino acid on a different part of the polypeptide, which makes that polypeptide is going to hold together in that three-dimensional structure. Those can be ionic bonds, those can be hydrogen bonds, they could be hydrophobic attractions between parts of amino acids that are nonpolar. There's a variety of things that will hold that three-dimensional structure in place. Now, at this point, we've covered primary structure, secondary structure, and tertiary structure. And those three levels of structure apply to individual polypeptides. But we're not quite at a protein. Not until we start talking about our fourth level of structure, which is the quaternary structure. Now, it is possible. In some cases, a protein might be a single polypeptide, and there are a few out there. So once I got my tertiary structure, that polypeptide, on the basis of its structure, could begin functioning as a protein. But the far majority of proteins are compositions of multiple polypeptides. When you put multiple polypeptides together, and remember, each of those polypeptides has a specific shape, and each of those polypeptides has a specific amino acid composition. If I put those multiple polypeptides together, what I get is a really, really big, really, really complicated three-dimensional construction, which is my full-blown functional protein. If you take a look at this illustration here, the implication is the protein at the bottom of the illustration is a composition of two polypeptides. If we were talking about hemoglobin, there would be four of them there. Four polypeptides, each with their individual primary, secondary, tertiary structure, all grouped together into one big three-dimensional protein with a very, very specific shape, and as a consequence, a very, very specific function. So obviously at this point, I've been harping about structure, and I've been harping about shape, I'm harping about function. Proteins are arguably, I had said this once before, the most important molecules to us. Perhaps even more important than carbohydrates, perhaps more important than fats. Aside from the fact that carbohydrates and fats get us calories, proteins are literally the molecules that provide for our life. Proteins determine what we look like, how we function, and how we act. There isn't a single functional aspect of our life that doesn't involve a protein in some way, shape, or form. And where the rubber hits the road, why do you think proteins are encoded by our genes? Obviously, if our genes are instructions for who we are, how we are, and what we are, proteins, which is what our genes code for, are going to be critical to who we are, how we are, and what we are. So perfect structure equals perfect shape. In other words, if there's something wrong with primary structure, there's going to be something wrong with secondary structure. And there's probably going to be something wrong with tertiary structure, which means there could even be something wrong with quaternary structure. Any problem with structure is going to negatively impact the function of that protein. Hemoglobin, its ability to bind oxygen depends on that perfect shape. mess with that perfect shape, you mess with the perfect function, and hemoglobin no longer binds oxygen. So I already mentioned that the instructions for making proteins are found in our genes. You remember that a gene is a sequence of DNA that codes for a protein. I promised you once upon a time that would not be the last time we saw it, and this will not be the last time we see it either. We understand that proteins are really important to life, and if they are so important, perhaps we want to regulate them. And perhaps the best way to regulate them would be at the level of the gene. If the gene contains the instructions for building the protein, there's a number of ways we can actually control whether that protein is in fact made or not at the level of the gene. Let's take a look at two illustrations on our slide. The top right, you'll see one illustration on the left showing you packaged DNA. And the packaged form of DNA on the left is labeled euchromatin, and the packaged form of DNA on the right is called heterochromatin. And then there's the word OR. And next to that, a couple illustrations showing me a gene that is stylistically illustrated with an on-off switch. We'll talk about that in a moment. Take a look at our bullet points. If proteins are so important to life, they must be regulated. How? All right, well, genes can be turned on, and there's two ways we can actually effectively turn a gene on. The first is how that gene is packaged. The gene is a sequence of DNA that codes for a protein. If I can access that gene, if I can actually read the information in that gene, I'll be able to make the protein. If you take a look at the illustration that's labeled euchromatin, you will see that the euchromatin or the euchromatic DNA there is packaged very, very loosely. And as it is packaged very, very loosely, I could effectively access or read that information. So you'll see the word euchromatic, which is essentially nothing more than an adjective corresponding to the idea of euchromatin. This is a loosely packaged form of DNA that is readable and therefore can result in the formation of a protein. On the other hand, take a look at the other illustrations to the right of the word OR, and you'll see that in that case, if transcription factors are present, I can turn on the DNA or turn on the gene, so to speak, at which point it can be read and therefore a protein is made. In either event, whether I package it loosely or I have transcription factors present, I can at that point determine if I'm going to actually be able to make the protein before I'm actually getting in the process. So what if my gene's turned off? Well, at that case, I could turn off the gene before the protein's even made by packaging it much more densely. If you take a look at that first illustration, you'll see the word heterochromatin. In our bullet point, you'll see the word heterochromatic, which once again is nothing more than an adjective to describe a highly, densely packaged form of DNA that is, for all intents and purposes, inaccessible. It's packaged so densely I could not access that information, and because I cannot make the protein. To the right-hand side, the other way I can do it is just having transcription factors absent. In a nutshell, transcription factors, and I guess I should have said this earlier, transcription factors are nothing more than a series of molecules that allow a piece of DNA to be read. And if you can't read it, you won't make the protein. So in either event, based on my packaging, if it's loosely packaged, I can read it, I'll make the protein. If it's densely packaged, I cannot read, I can't make the protein. Or if transcription factors are present, I can make the protein. Or if transcription factors are absent, I cannot make the protein. There's one other set of bullets here, and that is, can we regulate the protein after it's made? And that is a very real possibility. I mean, one of the ways we could do it is just simply change the shape of the protein. if you change the shape of the protein, you alter its ability to do its job. It's kind of a way of increasing the activity of the protein or decreasing the activity of the protein based on changes in its structure. You will see the word denatured under the bullet point. You will see the word renatured under the bullet point. Denaturing a protein, a process called denaturation, is creating a negative change in protein structure. As a consequence, the structure is altered and the protein does not do what it's supposed to do. On the other hand, you could bring that structure right back to where the protein has the exact same activity as it had earlier. If you take a look at the illustration at right, you will see a normal protein, and you'll see its denatured form, and you will see the denaturation arrow indicating the process of denaturation, and you will see an arrow indicating the process of renaturation. And since I didn't mention it specifically before, I suppose I should probably say that renaturation is a positive change in protein structure. Now, there's one other point I need to make, and it's not specifically mentioned on this slide, but it's this. In some cases, denaturation can be an irreversible process. Biologically, in most cases, denaturation and renaturation are normal composites of one another, and so you can have an endless process of denaturation and renaturation as a means of regulating protein activity. However, there are some situations where extreme heat, extreme ions, extreme acidity can create a denaturation event that will render a protein so denatured that it will not be able to be renatured. Case in point is when you cook an egg, you know that the goopy clear part of the egg, which everyone calls the white, technically is a protein called albumin. And when you subject that albumin to high temperatures, it becomes denatured. That's the process of taking that goopy, liquidy, white portion of the egg becoming this rubbery, hard, white stuff. And you cannot get it back once it's that point. I had mentioned earlier in this presentation that these molecules, carbohydrates and lipids and proteins, are all sources of energy. But the thing is when you are confronted with which food molecule to use as a source of energy, carbohydrates are preferred, fats come in close second, and proteins are a distant third. Proteins are a last resort source of energy, primarily because proteins are so critical for many other important things. For example, in the human body, proteins are an important constituent of our muscle tissue, they're an important constituent of our bone structure, and you can't live if you don't have a significant amount of muscle tissue. If you start breaking down proteins because you need a source of energy, you at some point are going to die. So proteins are last resort of energy, although I should mention that the amino acids that make up our proteins can be used as a source of energy under certain circumstances, and we'll see that in a little bit. But let's talk about protein starvation. The starving individual who doesn't have enough calories to sustain life, really their body has no choice but to start breaking down proteins. And the threshold of that breakdown is if you get to 50% of protein stores being lost, at that point, death will occur. The disease state that arises because of protein malnutrition is called marasmus. That's the person who doesn't have enough calories to sustain life and at the same time does not have enough protein to sustain life as well. So the obvious conclusion is that if you're starving, you can actually break down your O protein as a source of energy, even though it's the last resort. So let's talk about that process. The catabolism of proteins can yield amino acids. Amino acids are carbon-containing, energy-containing molecules. So these amino acids can be converted into a form that can ultimately enter cellular respiration to make ATP. We haven't covered cellular respiration in detail, but if there's some way that I can inject some energy-containing molecule into the cellular respiration process, I can generate some ATP from that molecule. Now, the process of using these amino acids to do that is called gluconeogenesis. We're going to see in a little bit that what we can do is take an amino acid. As a matter of fact, I can take 3 amino acids. I can manipulate them in such a way that those 3 amino acids can create a brand new molecule of glucose that can then enter the cellular respiration process. So if we take a look at that term, gluconeogenesis, it literally means creation of new glucose. Put it another way, we're making glucose from amino acids. So let's take a look at how we can actually manipulate these proteins so we can yield amino acids and then manipulate those in order to make these energy-containing compounds that can plug into cellular respiration. Well, when you break down proteins, you hydrolyze those peptide bonds. Remember, a peptide bond is a linkage between amino acids. Hydrolysis is the breaking of that. So if I'm going to hydrolyze a protein, I am breaking the peptide bonds and I'm yielding amino acids. In order to use the amino acid as a potential energy source, I need to remove the amino group. That process is called deamination. If you take a look at our illustration at right, you will see an amino acid, you'll see an arrow labeled deamination, and you will see the amino group being removed. At this point, the amino group becomes a waste product. It could be converted into ammonia, but that's terribly, terribly toxic. But instead, what our liver does is it converts that NH3, that amino group, into an organic compound called urea, which is ultimately removed from our bloodstream by the kidneys and is excreted from the body as urine. So what we have left over, though, is this 2-carbon compound, and you'll see the other arrow pointing to it, and you'll see the side chain labeled R, and then you'll see the 2-carbon compound, and that particular 2-carbon compound can be plugged into cellular respiration. Or what I could do is take 3 of those. And if I take 3 2-carbon compounds, that gives me 6 carbons. That gives me a molecule of glucose that could likewise be plugged into cellular respiration. That's where we get the term gluconeogenesis. The resulting 2 carbons can plug directly into cellular respiration, so I can make ATP to stay alive. The amino group is converted into ammonia, then into urea, and is excreted in the urine. All right, 2 more slides about protein. We're going to talk about protein quality in the diet. Human diet requires protein, and protein is really important because it maintains muscles, it maintains bones, it gives us the enzymes we need to be able to metabolize things. Protein is a really, really big deal. So when you consume foods, you want to make sure that the protein in those foods have the significant amount of amino acids that you need, because you're going to need those amino acids to build your own proteins. High quality proteins are proteins that contain a significant amount of those essential amino acids, and those proteins, as a rule, come from animal sources. On the other hand, low quality proteins are generally of plant origin, and they usually have fewer to no essential amino acids. So therefore, from a practical standpoint, the person who eats meat has no problem supplying themselves with sufficient essential amino acids to maintain quality of health. On the other hand, vegetarians, vegans in particular, have to be very, very careful in terms of what they consume to make sure they get those essential amino acids, since they're operating at a deficit, since they're not consuming animal material. You still need those essential amino acids. The good news is that through judicious, wise diet choices, a vegan can in fact get those essential amino acids without having to resort to consuming animal proteins.